right. So we are going to practice everything that we've learned by looking at some cells under a microscope. And so we're going to do an online lab that we've done for several years now. It's a good lab. They've done a really nice job with it. So what you're going to do, this is just a video explaining how you're going to do this lab. Let me go ahead and click on the link, and that's what you're going to do as well. You're going to click on the link and you're going to get to this page called the Biology Project Online Onion Root Tip. Now, the nice thing about this lab is it will not let you do it incorrectly. If you get it wrong, they politely tell you so, and then you just try it again. So here's what you're going to see. Now, you can zoom on your Chromebooks if you need to. For whatever reason, they make these really tiny. And so they're going to give you little pictures of an onion root tip cell. And this onion root tip cell is in a certain stage of the cell cycle. And they can be either an interphase, prophase, metaphase, anaphase, telophase. Now, let's take a look at this one. And the first thing that we should notice is all of the chromosomes are lined up in the middle. And that is a huge hallmark of metaphase. Now, what you're going to do is click on the name, and yay, they give you a handy dandy little chart. And notice they put it in the chart in the correct place. Yay. Now, you're going to keep doing this. Let's do another one together. And this one, the thing to notice is we've got a very well-defined nucleus and a well-defined nucleolus. Some students want to say prophase because that white area, but you don't see any specific chromosomes. This is still a nucleus. So let's go ahead and put it in interphase. And then, yay, they've done it for us. Now, let me show you what happens when you do it incorrectly. And then you're going to do it yourself. This is just a little tutorial for how to do this. Now, let me show you what happens if you do it incorrectly. Now, you can see this looks like a cell in interphase. But just for the sake of showing you, let's say you pick prophase. It's going to come back and it's going to tell you politely that you did it incorrectly. This cell has a prominent nucleolus, which is not a feature of cells in prophase. In prophase, the chromosomes are condensing and the nucleolus disappears. Try it again. And you click try again, and you're back to where you were. So then you click, oh, look, that's another interphase. And there you go. It puts it in. Now, you're going to do this for quite a bit. It'll take you about 4 minutes to go through them all, maybe 5 minutes. And then you're going to count them all up. You're going to get them all in a nice column. You're going to count them up. And then you're going to go back to our chart, and you're going to put the number of cells in each box, and then you're going to count them up to get a total. And then you're going to find the percentage. Now, percentage is something that you guys have done in previous math classes. And so I don't think it should be too outrageous. And let's take something that you might be familiar with. If I asked you to find the number of boys in the class, you might then look around your class and go 1, 2, 3, 4, 5. Okay, I've got 7 boys. And you notice that there are 20 students altogether. To find the percentage, you would just take 7 over 20 and then multiply that by 100. And then that would be your formula for percentage. So you're going to count up the number of cells in interphase, and then you are going to divide it by the total number of cells, and then multiply that by 100. And that will give you your percentage. And then you're going to try your hand at making a graph. Now, let's go ahead and go to the next slide. And we've got our spreadsheet. Now the spreadsheet's going to make you, make a copy. Make a copy. I'm spinning around a little bit. And here we have our, okay, you're still loading. Now, just for the sake of argument, I'm going to just put some fake numbers in here. Let's just make one 15, another one 25, and let's just go back and do 20 and then 20. Now, remember what you're going to do is copy both rows and both columns, and then you're going to go over here and click Insert Chart. It's going to spin around, and there you go. It's going to make a lovely chart for you. If it gives you a column chart, all you got to do is go to the chart type and hover over it, and it'll tell you a suggested, but you can also then say, oh, I want this and want this. So you should be able to find pie chart in there. Just scroll down. There you go. You can click pie chart. If you want to be fancy, you can pick this one, or you can pick the other one. You can have a little fun with it if you want. But then you're going to copy your chart right there, copy, and then you're going to go over and then insert your pie chart here. And that will be what you need to do for your data. And we'll talk about what this data means. And you're going to insert the pie chart. And then you're going to wrap it up by doing a couple of do we get it slides. One with, this is animal, these are animal cells, and then with plant cells. So again, you're going to practice these just to really hammer home. So what you need to do to turn this in is complete slides 29 through 33. That is your mitosis lab. There's no conclusion for this right now. We want to do the data. And so you should, by the end of this class period, you should have your Jamboard done and at the very least your mitosis lab started. So hopefully that gives you a good way to get started.